Meanwhile, a 17-year-old boy in the US has developed a tool that diagnoses autism and ADHD with 89% accuracy that simply involves taking a photo of your retina.
89%, 90%, 100%. Three numbers, one story, a 17-year-old, a photograph of your eye, and a machine that can supposedly tell whether you're autistic. So apparently I could have just skipped the waiting list, the assessment, the questionnaires, the interviews, all the money, all of it. I just had to look into a camera and let a laptop have a guess. By the end of this, you'll understand why 100% accurate isn't the good news that it sounds like. In medical screening, saying 100% accurate is the phrase that should make you go and find out what's been broken. So grab yourself a cup of tea, acquire a cat, or a plushie will do. This one has got more layers than a 4-month-old onion forgotten at the back of a fridge. If you're new here, hello, I'm Mike, a late identified autistic adult, formally diagnosed at the tender age of 42, which means for 42 years I was just a bit much, not living up to my potential, lazy, whatever, until a team who actually specialise in autistic adults looked at the evidence and went, ah yes, that. This channel is Autistic AF, evidence-based autism content, and roughly 16 cats, none of whom have consented to a retinal scan, and yes, I have thought about it, and they know. Here's why I'm the wrong person to sell the story to. Before YouTube, I was a software engineer, and for about a year I was the technical lead on systems for the NHS diabetic eye screening programme. I was not a clinician, I did not diagnose anyone, and I did not run the screening programme. I was tech lead, that means I led a software team that engineered a solution to handle retinal imaging, that's retinal photographs, eye pictures, at population scale. Fundus images of the back of the eye are used to catch diabetic retinopathy early in people who already have diabetes. The photo decides who needs a closer look and automatically refers them for an appointment. It does not diagnose and it does not replace the doctors. So when a story goes viral saying that we can read a brain from a photo of the retina, I'm not watching this as a bloke with a ring light and some strong opinions. And I'm also not watching as a doctor. I'm watching as the engineer who spent a year in the plumbing of a real national screening system, learning from the medics what a retinal photograph is allowed to claim and what it's not. Anyway, some of you have followed my eyeball odyssey. I lost most of the sight in one eye over about 5 months this year. I then had surgery and spent an unreasonable amount of time over the last year with people shining lights into my eyes and taking pictures of my retina. In fact, this here is my retina. So, retinas and me, we're close. The full saga is linked in the description below if you're interested, but come back though, I need you here.
Early diagnosis and intervention are critical for improving outcomes in autism spectrum disorder, or ASD, as well as ADHD.
The researcher is Edward Kang, and for the 2026 Regeneron Science Talent Search, which is one of the most serious school science competitions on the planet, he came second. And that is a real achievement. This video is about the claims that travelled, not about Edward as a person. Edward didn't just train a machine learning classifier. This year, he tried to open the black box, generating saliency maps to see where the model looks. And he went into the wet lab, growing retinal cells and running gene expression analysis to ask why there might be a retinal signal at all. Computational and biological in the one project, that is a lot of range and it deserves credit for his pure ambition alone. The underlying idea isn't new either. The retina is not just a camera part. Developmentally, it's an outgrowth of the central nervous system. It's the one piece of your brain a clinician can photograph directly without opening your skull. So the notion that neurodevelopmental differences might leave some trace there is a legitimate, live, active research field. And the problem he's aiming at is real and it is brutal. On the NHS, an autism or ADHD assessment can mean years on a waiting list. Some areas have closed their lists entirely. If you go private, it's hundreds, often thousands of pounds, and it might as well be $1 billion American for all the chance many people have of being able to access an assessment. When somebody looks at the current systemic healthcare problems and reaches for anything faster, I understand that reach completely. The project is ambitious, the field is legitimate, the access problem is a scandal. Now, let me show you why I'm filming this video for the second time. Here's what the internet decided RetinaMind was.
This is reporting from the Smithsonian Magazine and the Society of Science, and the original idea actually dates back to the Chinese University of Hong Kong that first linked retinal images to autism. The work is genuinely impressive.
And the reaction was enormous, because look what it's promising against the current status quo. A cheap, instant, objective eye scan that can skip the whole assessment nightmare.
Unfortunately, current diagnostic methods rely on multi-hour and subjective psychiatrist evaluations that are impractical for screening applications, ultimately resulting in costly delays for patients. Instead of only looking at behavior to diagnose these conditions, I explored the retinas of our eyes as an objective and efficient screening biomarker for neurodevelopmental disorders.
Of course, that went everywhere. So I assumed the press invented the word diagnose. So I went to the source to prove it, and I want to walk you through exactly what I found. I started with the most careful source, the official finalist profile. It calls RetinaMind a screening tool, a prototype, built to show how it might work. No accuracy figures given, none. And if every source read like that, I wouldn't be here. That is a fair, measured description of a student science fair project. But then I opened Edward's competition poster, and the poster's own title describes, and I quote, a method to diagnose neurodevelopmental disorders using the retina. A heading reads, enabling retina-based diagnosis. The results box says, 89% accuracy in autism and ADHD diagnosis. Another line says, RetinaMind could help diagnose ASD and ADHD earlier. The word that I was thinking the press invented is right there in the student's own poster, more than once, welded straight onto the number 89%. Smithsonian attributes that to him and quotes him describing how the diagnosis with the highest confidence becomes the official diagnosis of the model. So I take my first version back. 89% is the figure, diagnose in places is Edward's words. But the Society for Science's own social media account posted that RetinaMind accurately diagnoses neurodevelopmental disorders 89% of the time. That is the hinge between a careful project and a viral headline, and it's an institution standing on it. This is how internet myths are born. The qualifiers just fall off. You know, prototype, gone. Screening tool, gone. Methods not shown, definitely gone. And the 100% accuracy figure floating around, that one genuinely isn't Edward's at all. Let's take the 89% first, because it's the one people think that they're quoting. Can we check it? No. There are no public methods. We don't know if it's one model sorting 3 groups, or several models, each sorting 2. We don't know the class balance. We don't know whether one child's images could sit on both sides of the split. We don't know the sensitivity. We don't know the specificity, the confusion matrix, or whether it was ever tested on a fresh data set that it hadn't seen before. None of that is public, and a single reported accuracy figure with no methods is not something anyone can stand behind. I'll note this was never published as a peer-reviewed clinical study. It was reviewed by competition judges, not peer review. And there's one more document worth reading. Every project at this competition files an official certification form, and this one is Edward's. On it, the box for human participants is not ticked. What is ticked is tissue and potentially hazardous biological agents. This is the cell culture and the valproate. It's signed off as a continuation of last year's project and marked as not carried out in a regulated research institution. The form doesn't spell out the methods, and I'm not saying it should, but it is completely consistent with what the rest of the materials describe. A model working on existing retinal images and cells grown in a dish. What there's no sign of anywhere in the public record is the thing a diagnostic claim would actually need to support it. What would a diagnostic screening tool need? What methods would it need to validate it? Well, it would prospectively need to recruit autistic and non-autistic children or adults and run a controlled screening trial on them. On the evidence that's public, doesn't appear to have done that. That's the 89%. It is a claim made without evidence that is impossible for us to verify. So what about the 100% accuracy figure? To be clear, that's not a RetinaMind result. It comes from a separate South Korean study that Edward cites, and technically it didn't report 100% accuracy. It reported an AUROC of 1.0. That is a different measure. In plain English, give it one autistic image and one non-autistic image from that test data, and it will rank the autistic image higher every single time. Online, of course, they both become 100% correct because apparently statistics hasn't suffered enough. I'm not saying anyone deliberately swapped the numbers, but once a perfect score enters a story, it sticks. It sounds good. And in medical imaging, a suspiciously perfect score usually means the machine has found a shortcut and there's a problem with your methods. It doesn't mean you've found a cure or you've got a good result. 100% is not good. About 6 years ago, researchers had an AI that was very good at spotting melanoma from photos of moles. They took harmless, benign moles and then drew next to them that little purple marker line that surgeons often use to flag a lesion. They changed nothing about the moles themselves. They just added a pen mark. And the AI's confidence that these harmless moles were cancer shot right through the roof. And its ability to correctly clear a healthy mole fell from around 84% confidence to 46%. It turns out that this diagnostic AI had not learned to spot cancer. What it had learned is that when a doctor bothers to draw on a mole, it's probably worrying. And that's why it was flagging those images. The AI had learned the pen, not the subtle differences between a normal or a cancerous mole. The same thing also happened with COVID chest x-rays, where AIs were found to be scoring which hospital scanner took the image, not the disease itself. This effect has a proper name in computing. It's called shortcut learning. The model grabs the easiest thing that separates the two piles, and the easiest thing is very often nothing to do with the illness or what they're looking for. Back to the eyes. When a retina model scores 100%, the first question from anyone should be, what's different about these two piles of photos that has nothing to do with autism? And in the Korean data, there is a candidate the size of a bus. All of the autistic children in the Korean autism group were photographed fresh in one window in 2022. The comparison children's images were pulled from hospital archives going all the way back to 2007. So the AI might not have been asked autistic or not, it might have been asked photographed in 2022 or on a 15-year-old camera. And a computer can tell the difference between a 2022 sensor and a 2008 one perfectly. That's not neuroscience, right? That is a photograph's date of birth, in the same way that that's a marker pen, not cancer. And that's just a different x-ray machine. It's a different camera, not long COVID. And on top of that, the model kept its perfect score using only about 10% of the image, basically just a bit of the optic disc. And the uncomfortable connection for Edward is this is one of the exact regions Edward's own heat maps lights up on.
I started by generating in silico saliency maps that reveal where my model is looking on a retinal image to make any given diagnosis. Generating these visualizations for many images revealed 3 specific retinal regions as crucial for diagnosing ASD and ADHD: the fovea, optic disc, and blood vessels.
Maybe that means there is a real signal concentrated on the fundus imaging just there. There really could be. Or maybe a technical fingerprint lives in that same patch, and without external validation, a perfect score genuinely cannot tell you which is which. There is another confounder here too, which is that autism and ADHD often co-occur. In fact, they co-occur more often than not. If you're asking the AI to tell autism and ADHD apart, you're going to have a bit of a problem. Plenty of people meet the criteria for both autism and ADHD at the same time in the same body. So a model that picks a single winner from 3 boxes, lovingly referred to as normal, or autism or ADHD, whichever scores the highest, and the Smithsonian quote about the official diagnosis of the model suggests exactly that kind of winner takes all. Actually is answering the question the clinic doesn't ask. Can the model hold both at once? Is it 3 mutually exclusive boxes, where a confident autism score quietly buries a legitimate ADHD one? The public materials don't tell us, but a system that has to pick one is already a vast oversimplification of the thing it claims to detect before we even get to accuracy. To Edward's real credit, he saw the black box problem coming. Last year's model, in his own words, was completely black box, so this year he tried to open it. First, the heat maps. He generated saliency maps, pictures showing which pixels pushed the score, and found the model leaning on the fovea, the optic disc, and the blood vessels. That is more thoughtful than most projects ever manage. But here's a subtle problem with these maps. A saliency map tells you which pixels moved the score the most under one particular method. It does not tell you why they mattered, and it does not prove that there is an autism-specific signal in them. Remember what we said before about the AI seeing a doctor's marker pen and leaping to conclusions? The shortcut can live inside the highlighted region. A heat map glowing on the optic disc is equally consistent with a real biological signal, but it's also equally consistent with a scanner fingerprint sitting over the optic disc, or a Canon camera instead of a Sony one, for example. The picture looks like an explanation It looks cool and Star Trek-y, but it isn't a validation. A model you can't see inside might be doing something real, or it might be doing something silly. And the whole point is you can't yet tell which is which. Calling it arbitrary and then clinically powerful in consecutive breaths is this two voices problem again. Second, the super ambitious bit, the Petri dish. Edward built what he calls the first retinal cell model of autism.
To validate these results biologically, I created the first ever in vitro retinal model of ASD using a retinal cell line and a well-known autism-inducing chemical called valproic acid.
He took a retinal cell line and then dosed it with valproic acid, and then measured which genes changed. Valproate isn't a random pick. Taken during pregnancy, it is associated with a raised chance of neurodevelopmental differences in the child. And off the back of this, researchers dose pregnant rats and mice to make what's called the VPA model. Animals that show some autism-associated behaviours. That model is genuinely useful in some ways. That model is also already one step removed from autistic people. We are not rodents. It's a chemically induced proxy in a rodent, and how well it stands in for human autism itself is argued over. I mean, autistic people not being rodents, for one. Now watch what the experiment does carefully, like it's a magician doing a card trick, because it is kind of similar. It doesn't even use the animal. It takes that same chemical and puts it on retinal cells in a petri dish, and then reads which genes change. Those are the rodent retinal cells being exposed to the chemical. So we're now on a proxy of a proxy. Not an autistic person, not even a rodent behaving in autistic-like ways. Believe it or not, that is a real area of study. It's as batshit as it sounds. Cultured cells reacting to a drug. And we have to be exact about what dosing cells in a dish shows. In his talk, he calls valproate
A well-known autism-inducing chemical called valproic acid.
a well-known autism-inducing chemical. And that phrase does a lot of quiet work. Pouring valproate on retinal cells does not make those cells autistic. It produces a chemical response being used to stand in for one narrow proposed pathway. A cell in a dish reacting to a drug is a model of a mechanism. It is not a rodent, it is not a person, it's not autistic behaviours or traits or features, and it is certainly not a diagnosis or even a screener. That experiment, at its best, is a lead. A potential clue, a hypothesis for someone to chase. What it's not is evidence that the photo classifier separated its images because of those genes. Nobody has shown that link. It also doesn't show that autistic people's actual retinas carry these changes because no autistic tissue was ever tested. It was cultured cells reacting to a drug. So the poster says the findings, quote, support a genetic foundation for retina-based diagnosis. I have to push back on that hard. On the science, again, not the person. Edward's very clever, very ambitious, still a legal child, still inexperienced, but it's the science, it's the ideas, not the person. Just please, okay, before anyone goes wild in the comment section. Genes changing their expression when you add a chemical is not evidence of an inherited genetic cause. These are two different claims, and the gap between them is enormous. The accurate, but still interesting version is, you could say something like, I don't know, we found retina-related genes that respond to valproate. Worth studying, maybe? I mean, you know, genetic foundation for diagnosis is a really bigger claim, right? That is like a check that his experiment can't cash. So let's grade Edward's wet lab work. As a cell experiment, interesting lead. As evidence for a genetic autism test, or as validation to support his eye scanning AI, it barely crawls out of the petri dish. I'm struggling to award it anything above an E tier. Fortunately for Edward, Regeneron's judges and their bank account are considerably more generous than me. Let's set all of that aside. Let's be maximally generous. Let's pretend that 89% is rock solid. Let's pretend that these claims are real and they are true. First, we have to test that claim against two very simple ideas. Firstly, sensitivity. How good is the test at catching the people that genuinely have the thing you're searching for? And specificity. How good is it at correctly clearing the people who don't? We could say this 89%, which is a figure pulled out of somebody's arse, but let's give it that figure, let's call it something. Let's say it's 89% on both of those things, sensitivity and specificity. In the academic literature, well, I'm not going to talk about what Mike currently thinks or Mike's hot takes or what you might think. The literature is currently sitting at around, at least at the time these studies were done, roughly 3 in 100 prevalence rate. Okay? It's almost certainly more than that, but let's leave it there. When a thing is that rare, even a good test will throw out a mountain of false alarms. If you screen 100 children, where only 3 are truly autistic, and of everyone the test flags, only about 1 in 5 is actually autistic. The other 4 in 5 get false alarms. That's not controversial, it's just how rare things behave. What happens when somebody asks an AI, we're getting a bit meta here, to fact check the claim? A user on X, formerly Twitter, asked Grok, "Is this accurate?" And Grok says, "Yes, this is accurate." Then in the very same answer, it admits that RetinaMind is a research prototype using public data sets, that it's not clinically validated, it's got no working demo. So what exactly has it verified? That the project exists? Not that the 89% is reliable, but yes is the bit people remember. Grok is misleading here. And just like many LLMs, it's just repeating the headlines. This is how internet myths are born. So much for a maximally truth-seeking AI, Elon. Even being generous with its sensitivity and specificity, it'll be throwing out mountains of false positives. And a false positive is not merely a wrong square in a table if the result moves a child towards treatment. Even a correct autism flag does not tell us that the child needs to be treated, trained to look less autistic, probably. Follow the arrow on Edward's own poster. Patient, screening, diagnosis, treatment. This is the medical model. The project talks about earlier diagnosis leading to earlier intervention and better outcomes. It never defines the crucial words. What intervention follows the flag? What outcomes are supposed to improve? Who chooses them? Does the support build communication and autonomy, or does it train the child to suppress the traits that made them visible in the first place? And this is important because early intervention is not just one thing. Some of it is genuinely good, and I want to name it clearly so it's not heard as support is bad. Communication access, sensory accommodation, occupational and speech support, offered but not imposed, I should say. Informed families, schools that bend the environment around the child instead of bending the child. That kind of early identification followed by affirming support can change a life for the better. But the elephant in the room, in the United States especially, early autism intervention very often means intensive behavioural programs, and I've spent a whole other video on the evidence and the ethics around those, so I won't relitigate it here, but on screen and in the description. I'll say only this right now, carefully. Nothing in Edward's materials say he endorses any particular therapy, and I'm not going to put words in his mouth. The problem isn't his intent. The problem is that a screening pathway is only as good as what it signposts you to next. It goes, you're in, right, over here, follow that door over there marked with the letters ABA on it. And earlier does not make an undefined intervention automatically kind. A tool that flags more autistic children, 4 in 5 of them wrongly, into a treatment system no one has specified, is not self-evidently a gift. Thank you, Edward. So let me put the pieces together. Any screening test flags you, it does not diagnose you. And that is true also of questionnaire-based screeners like the RADS-R or Autism Quotient AQ tests, and it's true of biomarker prototype screeners like this one. To earn even that narrow job, a screener has to be validated in the very population that it will be used on with known sensitivity, known specificity, and a real assessment on the other side of the flag. And that is exactly what the study we established is missing, the prospective trial in real children that the public record gives no sign of. In diabetic eye screening, the photo is a signpost to a specialist who examines you properly and decides. It's never the diagnosis. And even to be a signpost, that went through years of validation and national standards and clinical controls. And I know because those are the rules that the software has to respect. RetinaMind, and above all the noise around it, skips every single one of those steps and inflates the whole, hey, this photo has an interesting pattern to it, to this photo tells you are autistic, which no one has earned the right to say. Not Edward, not Yonsei, not Smithsonian, not yet, and maybe never from a photo alone. Notice who's absent from the entire story. Every one of these tools measures autism from the outside, from a photograph, from what a machine can observe about you. At no point does anyone ask the autistic person about their life. We have built machines to detect us and forgotten to include us. It's there in the language the whole pipeline runs on. Patience, pathophysiology, diagnose then treat, a clinically powerful tool, normal as the other option. That's the medical model start to finish. And it's not a comment on a teenager's heart, it's a comment on the frame that he was handed, or that he grew up in, or that he's adopted. It's the same frame that the press and institutions and algorithms all reach for, because it's the frame that flatters a very quick answer. A retinal photo can, at absolute most, only ever be a signpost to a conversation. It cannot have the conversation, and the conversation, particularly by way of a diagnostic observational interview, that is a whole thing. And I've got a whole load of videos on all the various models of diagnostic interview. If this story feels familiar, it's because I've shown it to you before, with brains instead of eyes. Last year, Princeton University posted a study, a careful finding, also using AI, that autism might sort into 4 distinct subtypes. It was explicitly not diagnostic, but that didn't stop their press office saying that it paves the way for precision diagnosis and care. The news rounded it up. Social rounded it up even again. And what came out of that was, scientists found the types of autism. What subtype are you, says some content creators. None of this the scientists ever said. This is the same societal machine as before. A careful thing, maybe a bit less careful in Edward's early case here, but usually a careful thing goes in one end, and everyone downstream adds a little verb that the data can't support. And nobody who inflated it ever has to stand next to the exaggeration and stand in the public spotlight and defend their work. And that's the frustrating thing for me. Journalism seems to have lost so much of its integrity. But I won't go down that depressing rabbit hole today. The reason this story hits so hard isn't, school science fair project does a thing and it's not that robust. That's not really a story, right? The story is that the system is really broken. The waiting lists are real, the costs are real, the pain is real, the years of being told that you're just anxious or lazy or whatever, before anyone listens, is real, and it is a scandal. But the answer to a broken diagnostic system is not a camera that decides your neurotype for you, like some sort of digital version of the Harry Potter sorting hat. It's faster, kinder, properly funded human assessment with autistic people in the room shaping which questions get asked in the first place. I'm amazed. I'm amazed at the ambition. I'm also furious. Well done, Edward. I mean, if Edward carries on into medical research, I will be genuinely interested to see where the work goes after proper validation, external testing, and collaboration with autistic people. And I am furious that the machine that took a promising prototype and sold it as diagnoses you from a photo. I mean, have we learnt nothing from Elizabeth Holmes? No, I don't think we have. You know, like the social captions all the way up to an institution that should know better. If your dog just tilted its head at the screen, like it's just been shown a card trick, don't worry, that's the next video. Because apparently the dogs are portismly neurodivergent now, and I have some opinions on that. If this video helped clear something up, or helped something click, or maybe just left you with a ton more questions, stim the subscribe button if you haven't already, hit the like button, let me know your questions in the comments below, and I will see you